Hello friends, today we will discuss sterilization techniques. In the first part of sterilization techniques, we discuss methods of heat sterilization. In the same unit of paper 2, that is fundamentals of microbiology, we will discuss another concept of chemical methods of sterilization. So let us proceed with different chemicals used for sterilization purposes. So again, chemical sterilization is the process of removal of microbes or killing of microbes by use of certain chemical bactericidal agents. Certain chemicals that kill bacteria, use of such chemical is called chemical sterilization. We will discuss some certain important terms associated with chemical methods of sterilization. First important term is the disinfection. It is the killing, inhibition or removal of microbes or removal or killing of disease causing microbes. And this process or the disinfectants, they are usually applied to inanimate object. So disinfection is used for inanimate object, means chemicals which are used as a disinfectant, they are applied to inanimate objects. So it is called, rather than chemical sterilization, you can call it as a chemical disinfection. The better term will be disinfection rather than sterilization, because it does not always result in destruction of endospores of the microorganisms. It does kill vegetative cells but not necessarily endospores. Then another relevant term, antisepsis. Antiseptics, we better know this term. It comes from the word antisepsis. Sepsis means growth. So prevention of growth or prevention of infection, usually disease causing organisms, is called antisepsis. Then another term, bactericide. Bactericide is an agent that kills bacteria. Bacteriostatic is a chemical that does not kill bacteria, but it inhibits growth and multiplication of the bacteria. So these are important terms regarding chemical methods of sterilization. Disinfection, just now we discussed this concept. Disinfectant is a chemical used for disinfection, is called a disinfectant. We will discuss certain properties of disinfectant. Not all chemicals can be called as a disinfectant. It should possess certain properties. First property is that the disinfectant should have wide spectrum of activity, means it should be effective against variety of microorganisms. It should be effective against gram positive, it should be effective against gram negative. Likewise, it should have a broad spectrum of activity. Then second property, it should be able to destroy microbes within practical period of time. It should be speedy in action. It should rapidly kill the organism. Then it should be active in the presence of organic matter also. So organic matter should not alter the activity of a disinfectant. Then the disinfectant should be effective. It should make effective contact and should be wettable, means it should establish in contact with the object to which it is applied, so that it will be in direct contact with the microorganisms. Then it should be active at any pH values. It should not get destroyed by either alterations in pH, acidic or alkaline pH. Then it should be stable at wide range of pH temperature. And it should have long shelf life, means it should be storable for a longer period of time, and it should have high penetration power. These are the properties of disinfectant or properties of ideal disinfectant. A few more properties we will discuss. It should be non-toxic, it should be non-allergic, non-irritative and non-corrosive. Then it should not have any bad odor, otherwise it will become very difficult to apply these disinfectants to any object. Then after use it should not leave any residue or stain. Suppose you are applying it onto your hands, it should not leave a stain. Then its efficacy should not be lost on reasonable dilution. This is very important because many disinfectants are used only after dilution, but they should not lose their activity. Then last important property, it should be cheap. It should not be expensive. It should be cheap and easily available. So all these are ideal properties of a disinfectant. Now we will discuss some examples of the disinfectants mentioned in your syllabus. One of the example of a disinfectant, in fact it has been very widely used, alcohol. So mode of action of alcohol is alcohol results in dehydration. It removes water from the cell as well as it disrupts the membrane. It dissolves lipids in the lipids present in the membrane, so that it leads to formation of pores in the cell membrane, and through these pores the cellular contents are leaked out of the cell. So this is the mode of action. Even it results in coagulation of the proteins. Examples of alcohol include ethyl alcohol, ethanol, isopropyl alcohol, methyl alcohol. All these are widely used disinfectants. 70% aqueous solution is widely used as disinfectant. It is more effective than absolute alcohol. It is also called spirit. It is used as an antiseptic on skin. Then even isopropanol is also used. It is preferred to ethanol. It can also be used for disinfection purposes. It is used to disinfect thermometers. Then methanol, it can kill fungal spores. The disadvantages of this alcohol, it is a skin irritant. Then it is volatile, it evaporates very rapidly. These are the disadvantages. Second example mentioned in your syllabus, phenol. Phenol is again a very widely used disinfectant. The mode of action of phenol is that it acts by disruption of the membrane, or it results in precipitation of the proteins as well as inactivation of the enzymes. Proteins, which are major components of cell wall and cell membrane, they get precipitated, result in their deactivation. Even it results in inactivation of enzymes present in the cell. Widely used examples of phenol include phenol, which is used in 5%, and cresol 1 to 5%, lysol, and hexachlorophene. Chlorxylenol is well known, what we call as a Dettol. So Dettol is a well known disinfectant. It is a type of phenol. So even these phenols, they act as a disinfectant at high concentration and at antiseptics at low concentration. They are bactericidal, they are fungicidal, they are mycobactericidal. Means they can kill bacteria, they can kill fungi, and they can kill also mycobacteria, but they cannot kill endospores and virus, most of the viruses. Triclosan, chlorhexidine, these are widely used disinfectants among the phenols, out of which chlorhexidine can be used in isopropanol solution for skin disinfection purposes. Triclosan has a very good activity against gram positive bacteria. So this is how this phenol is another example of disinfectant. The disadvantages of the phenol include it is toxic. If it is used in very high concentration, it can cause skin irritation, skin corrosion. Then chlorhexidine is inactivated by anionic soaps. Even hard water diminishes the activity of chloroxylenol. These are the disadvantages of phenol. One more example mentioned in your syllabus is aldehydes. So these aldehydes cause alkylation of different amino, carboxyl or hydroxyl groups. Particularly, they damage the nucleic acids. They result in alkylation of nucleic acids. Even it kills vegetative cells as well as endospores. The examples of widely used aldehydes include formaldehyde and glutaraldehyde. So formaldehyde, aqueous solution of formaldehyde, 40% aqueous solution is called formalin. It is used for surface disinfection. It is used for fumigation. It can also be used in operation theaters, safety cabinets. Fumigation is achieved by boiling formalin. Even it is used in sterilization or disinfection of certain clinical equipments like thermometers, cystoscopes, bronchoscopes and centrifuges. The disadvantages of this aldehyde include it has poor penetration power, then its vapors in gaseous form, it can cause irritation, and its activity reduced in the presence of proteins. While glutaraldehyde requires an alkaline pH. These are the disadvantages of aldehydes. Ethylene oxide is another example of disinfectant. It is a colorless, odorless and flammable gas. The mechanism of this ethylene oxide is alkylation of sulfhydryl, amino, hydroxyl and carboxyl groups present on proteins and amino groups of nucleic acids. Even ethylene oxide gas is widely used as a disinfectant or sterilizing agent. It can kill bacteria. It is bactericidal, virucidal, even fungicidals. It also kills yeast and molds and is compatible with almost all materials repeatedly applied also. Then another example mentioned in your syllabus is beta propiolactone. It is a condensation product of ethane and formaldehyde. So it has rapid action and it is used in around 0.2%. It is more efficient in fumigation than formaldehyde. So it is used for inactivation of vaccines. So these are the different chemicals which are used for sterilization purposes according to your syllabus. In the first part we discuss methods of heat sterilization, and the second part we discuss today methods of chemical sterilization.